Hi everyone, welcome to chapter 10. So in this video, we're going to kind of go over a little bit more about experiments. So we've talked a lot about kind of variables and then a little bit about correlations. And now we're going to talk about experiments, which are distinctly different from correlations. And we'll explain how they can be used to actually draw some causal inferences or showing how one thing can cause a direct outcome on another thing. So let's go ahead and get into it. All right, welcome to chapter 10. So today we're going to talk about some simple experiments. And in this chapter, we're going to get into what actually makes a study a true experiment, look at some real world examples, and discuss how we lay out these designs. We'll also spend some time on how experiments give us the foundation to make actual causal claims and how we check those claims using our validities. All right, here's a quick overview of the main topics we'll be discussing today. First, I want to walk you through two examples of simple experiments that'll give us a solid baseline of what these are. From there, we'll look closely at experimental variables, specifically independent and dependent variables, but also control variables. Then we'll cover exactly why experiments support causal claims. We'll look at the differences between the two main ways to structure an experiment, independent group designs and within groups designs. And lastly, we'll wrap up by talking about how to go about interrogating causal claims with the four validities we've used before. All right, let's start off by looking at two simple experiments. We're going to look at an experiment that examined the effect of publicized norms to see if it makes a campus climate more inclusive. And then we'll look at a really neat study on how we go about motivating babies to keep trying when performing a difficult task. Let's look at the first example here. So researchers at the University of Wisconsin-Madison actually wanted to test the effectiveness of promoting a welcoming and inclusive climate just by changing the visible social norms around campus. They put up pro-diversity posters around campus, which publicized the message that the vast majority of students embrace diversity. They set up an experiment where some areas displayed these norms posted, while other areas displayed control posters, like the CDC flu vaccine poster that was on the previous slide. If you look at the graph, you can see a clear difference in the results. Students exposed to the control poster actually had a slightly negative inclusive climate score, while the students who saw the norms poster scored significantly higher in inclusion. This tells us that simply manipulating the visual environment can actually cause a change in people's perception of their community. Our second example here is a really fascinating study where they recruited more than 100 babies. They wanted to investigate whether observing an adult putting effort into achieving a goal would influence infants to try harder and persist at completing new tasks. In the effort condition, the babies watched an adult struggle a bit before trying until they successfully opened a toy. In the no effort condition, the adults just easily solved the task that they were doing right away without any real struggle. Then the babies were handed a totally new toy that had a simple button on it, but the button didn't actually do anything. The researchers measured how many times the babies pressed the button before giving up. As you can see in the chart, the babies who watched the adult put in effort pressed the button around 28 times compared to only about 17 times for the babies in the no effort group. So just watching an adult persist actually made the babies try harder themselves. All right, let's talk now about how experimental studies use variables in specific ways to draw these causal conclusions. When we talk about setting up a true experiment, we're looking at specific types of variables. Every experiment must have at least one variable that is manipulated by the researcher and at least one variable that is measured. We also look at things called control variables, which are the factors we intentionally keep exactly the same across the board. Let's break down these definitions now. The independent variable, or IV, is the one manipulated by the researcher. It's what the researcher has control over. The specific levels or versions of the IV are what we call conditions. For example, in our baby study, the IV was adult behavior, and the conditions were effort or no effort. In the poster study, the IV was poster type, with the conditions being the norms poster or the control poster. The dependent variable, or DV, is the variable that is measured, or your outcome variable. It depends on what happened with the independent variable. In the baby study, the DV was the number of button presses. In the poster study, it was the inclusive climate score. Now, there's also a third category called a control variable. Now, the name is kind of a misnomer because the word variable itself suggests change, but a control variable is actually anything that the experimenter holds completely constant across all participants. Why do we do this? Well, because we want to make sure the only difference between our groups is the independent variable itself. For example, in the baby study, they used the exact same room, the same background environment, and the exact same unpressable toy button for every single infant. By holding those things constant, we rule them out as potential explanations for our results. So why do we care so much about setting things up this way? Because experiments are uniquely qualified to support causal claims. If you want to say variable A causes variable B, you have to meet 3 specific criteria, which we've talked about earlier this semester. You have to establish covariance, you have to establish temporal precedence, and well-designed experiments must establish internal validity. Let's look at how experiments build all 3 of these into their methodology. Let's first talk about covariance. This basically answers the question, compared to what? As we've discussed, our everyday personal experiences don't usually have the benefit of a comparison group or a comparison condition. If you just guess that publicizing diversity norms works, you don't actually know unless you have a control or comparison condition to see what happens without it. Covariance is shown when the results of your groups are actually different, meaning the mean scores of your dependent variable shift depending on the level of your independent variable. When we talk about setting up these groups, there are a few types to keep straight. The control group is your neutral or no treatment condition. Keep in mind, not every single experiment needs a pure control group. Sometimes you just need a solid comparison group. The treatment group or groups are the active levels where you're applying your intervention or your manipulation. And a placebo group is a special kind of control group exposed to an inert or non-active treatment, often things like a sugar pill. The second condition that's necessary for a causal claim is temporal precedence. This just means the causal variable must happen before the effect variable in time. In an experiment, this timeline is guaranteed because of how we sequence the study. The researcher manipulates the independent variable first, like showing the baby the adult's level of effort, and then measures the dependent variable second, like tracking the baby's subsequent button presses. Because we control the timeline, we know for a fact which one came first. The third and often toughest criterion is internal validity. We need to be absolutely sure there are no alternative explanations for our results so we can confidently say the independent variable caused the change in the dependent variable. When an experiment is poorly designed, it runs into threats like design confounds or selection effects. Think about a confound as something that confuses your results. For example, look at the graph on this slide illustrating a potential design confound for our poster study. Imagine if the researchers put the norms poster in a waiting room that happened to have other students hanging out, but the control poster was left in a completely empty waiting room. Is the higher inclusive climate score because of the poster, or is it because there were other students in the room? Your independent variable is now confounded, and your internal validity is ruined. To keep your internal validity intact, you have to look out for systematic variability. This is one of the real problems. Systematic variability happens when an outside variable changes in a predictable way alongside your independent variable. Like if the effort group of babies consistently got tested by, say, a warm, smiling researcher, and the no effort group got a cold, unsmiling researcher. That would be a design confound. On the other hand, we have unsystematic variability. This is just random, haphazard noise that happens across both groups. If you look at the baby diagram here, you'll see kids who can't sit still, or kids who just had a nap, or instances where a parent might have looked away. As long as these random differences are spread out evenly across both conditions, it's not a confound. It can make it a little harder to detect your effect because it creates statistical noise, but it won't ruin your internal validity. Another threat to watch out for is a selection effect. This happens when the participants in one condition of your independent variable are systematically different from the participants in another condition. Look at the chart on the slide, which highlights a study on an intensive therapy for children with autism. One group got the intensive therapy and the other got the standard treatment as usual. The kids in the intensive therapy group showed way more improvement. Well, that sounds great, right? Except the researchers didn't randomly assign the kids. Maybe families who lived closer or families who specifically requested the intensive treatment ended up in that group. Because of that, the groups were systematically different from the start, and it's impossible to know if the therapy itself caused the improvement. The easiest way to avoid selection effects is simply to use random assignment. But what happens if your sample size is small? Random assignment can sometimes leave your groups a bit unbalanced just by pure chance. In those cases, researchers will often use matched groups or matching. Look at the graphic here. Say we're worried about participant characteristics like hometown size. We look at our sample, take the 2 people from the smallest hometowns, and randomly assign one to group 1 and one to group 2 using, say, a coin flip. Then we take the next 2 smallest and do the same thing all the way down to the 2 youngest. By matching pairs on a specific variable and then randomly splitting them, we guarantee that both groups are completely even on that characteristic from the start. Now let's look at how we structure experimental designs. There are 2 ways in which we can structure our design: an independent groups design and a within groups design. Let's break each of these down now. An independent groups design is when you have completely different groups of participants placed at different levels of your independent variable. So, if you're doing our poster study, a participant only ever sees either the norms poster or the control poster. They never see both. This is also called a between subjects or between groups design. A within groups design is when there's only one group of participants and every single person is exposed to all levels of the independent variable. If we did the poster study this way, every student would experience both poster conditions at different times. Let's talk more about independent groups designs. We'll break down some of the different ways we can structure and design these types of studies. In independent groups designs, you have 2 choices for how you time your measurements. The first is a post-test only design. This is really straightforward. You randomly assign your participants to your independent variable groups, you expose them to the condition, and then you test them on the dependent variable exactly once at the very end. You can see this mapped out in the diagrams on the slide. Participants are randomly assigned to either the norms poster or the flu shot poster. They look at the poster, and then they immediately complete the inclusive climate measure. The second option is a pre-test, post-test design. Here you randomly assign participants to your groups, but you test them on the key dependent variable twice. Once right before they experience the independent variable, and once right after. This graph uses verbal GRE and mindfulness study to illustrate this. Participants take a verbal GRE test as a pretest, then split into either a mindfulness class or a nutrition class for 2 weeks, then take another verbal GRE as a post-test. If you look at the graph, you can see exactly why this is useful. Both groups started at the exact same accuracy level on the pretest, but only the mindfulness group showed a statistically significant improvement at the post-test. So you might be wondering which of these 2 setups is actually better. Well, it entirely depends on the situation and goals of your study. Sometimes using a pre-test, post-test design can be problematic. For example, if your dependent variable involves something like physical exertion or eating food, testing participants twice might make them totally exhausted or completely full before the real post-test happens. On top of that, a pre-test can sometimes tip participants off to what you're studying. However, if you need to be absolutely certain that your groups are perfectly balanced from the start, a pretest does make sense. But don't underestimate post-test only designs. When you combine a manipulated independent variable with solid random assignment, it's already an incredibly powerful tool for showing cause and effect. Now let's switch gears and look at our second major category, within groups designs. We're going to cover the 2 main types, repeated measures and concurrent measures, and talk through the unique advantages and disadvantages of running a study this way. We'll also look at how these designs handle our causal criteria and clarify whether a standard pretest post-test design counts as a within groups design. So with a repeated measures design setup, participants are measured on the dependent variable more than once, specifically right after being exposed to each level of the independent variable. Here, let's look at a classic chocolate taste testing study. In this case, they brought college students into a lab, and in the first condition, a Confederate student is sat right next to them. They were both handed a piece of chocolate, tasted it at the same time, and rated how much they liked it without speaking. In the second condition, the Confederate left, and the participant tasted and rated a second piece of chocolate completely alone. The participants were told they were tasting two different types of chocolate, but they were actually identical. The graph shows that people liked the chocolate significantly more when they shared the experience with another person. This is a repeated measures design because the same person rates chocolate multiple times under different conditions. The other type of within-group setup is a concurrent measures design. Instead of having participants experience one level of the independent variable and then the next level in a sequence, they are exposed to all levels of the independent variable at the exact same time. And then we record a single behavioral choice as the dependent variable. Here we have a great example from Emily Bray's work on puppy behavior. A researcher pointed directly at one of two blue cups placed in front of a puppy. One was the pointed at cup, and the other was a control cup. The puppy is looking at both options simultaneously. The dependent variable is simply which cup the puppy chooses to knock over first. The graph shows that puppies successfully selected the correct pointed at cup roughly 70% of the time, which is way above random chance. In this design, they are proceeding all levels of the independent variable simultaneously and then making a single choice. There are three large advantages to setting up your study as a within groups design. First, the participants in your groups are perfectly equivalent because they are the exact same people. They serve as their own controls. And second, because you've removed all that individual background noise, these designs give researchers more power to notice true differences between conditions. Power is just a study's ability to detect a statistically significant result when an independent variable actually has a real effect. Third, these designs are incredibly efficient and require fewer participants. If you want 20 people in each condition, but you use an independent groups design, you need 40 unique individuals. But in a within groups design, you only need 20 people because each person is in both conditions. Even though within groups designs are incredibly powerful, we still have to hold them up to our causal criteria. Covariance is easy. We just look for a difference between the condition averages, but temporal precedence and internal validity can get tricky because of a unique threat called an order effect. When a participant experiences one condition, it can naturally alter how they respond to the next condition. To fight off order effects, researchers rely on a technique called counterbalancing. This means we split our participants up so that they experience the levels of the independent variable in different sequences. You can see this here where different groups taste the chocolate alone or with a confederate in different sequences or orders. Let's look a little closer at why order effects are such a threat to internal validity. If the order of conditions is what's changing the participant's behavior, then the order itself is a confound, and you can't prove your independent variable did the work. There are two main types of order effects. Practice effects. This happens when participants get better at a task simply because they've done it before, or they get worse at a task because they're getting tired or bored, which we call a fatigue effect. Carryover effects. This is where some form of contamination sticks around from the previous condition. If you did a study on caffeine and taste testing, but had the first condition be the caffeinated condition, there would likely be carryover effects on the second non-caffeinated condition, unless there was ample time in between each condition for the caffeine to leave the participant's system. The way we deal with order effects is through counterbalancing, and there are two ways to do it: full or partial. Full counterbalancing. This is used when you have a small number of conditions. You present every single possible order to different groups of participants. So with two conditions, there are only two orders: A, then B, and B, then A. Partial counterbalancing. What if you have several conditions? The number of combinations explodes, so we only use a select subset of the possible orders. A popular way to do this is with a Latin square design, which you can see represented on the slide. It's a structured layout that guarantees every single condition appears in each position at least once. Despite their strengths, within groups designs aren't perfect, and they have three main downsides. First, there's always a risk of potential for order effects, though we can manage that with counterbalancing. Second, sometimes a within groups design is simply not practical or even possible. Think about trying to teach a child how to ride a bike. Once they learn with method A, they're permanently changed, so you can't wipe their memory and test them again using method B. Third, within groups designs are highly vulnerable to demand characteristics. This happens when experiencing multiple levels of an independent variable gives participants enough clues to guess your study's hypothesis. Once they think they know what you're looking for, participants often start changing their behavior to match your expectations, which ultimately ruins your data. Now let's clarify a common point of confusion. Oftentimes students will look at pretest, post-test, and independent groups designs and think, wait, we're testing the participants twice, so shouldn't this be considered a within groups design? And the answer is no. Look at the two flowcharts on the side. In a true within groups or repeated measures design, every single participant is exposed to all levels of the independent variable. In a pretest, post-test independent groups design, a participant is still only ever exposed to one level of the independent variable. Testing someone twice on the dependent variable does not mean they experience multiple conditions of your independent variable. All right, let's head into our final major section, how we use our 4 big validities to evaluate causal claims. Whenever you are reading a study that makes a headline causal claim, you shouldn't just take it at face value. You need to actively ask yourself 4 questions. How well were the variables measured and manipulated? To whom or what can the causal claim generalize? How well do the data support the causal claim? And finally, are there alternative explanations for the outcome? Let's look at how we check each of these. To check construct validity, we look at both sides of our variables. First, look at the dependent variables. How well were they measured? For example, in our poster study, how did they calculate that inclusive climate score? Was it a valid and reliable scale? Second, look at the independent variables. How well were they manipulated? Researchers often use extra tools to be sure, like a manipulation check, which is an extra dependent variable inserted into the study just to confirm the manipulation actually worked as intended. They might also run a pilot study with a separate group of participants beforehand to test out the materials and make sure the manipulations work correctly before running the real experiment. Next, we have external validity, which looks at how well these causal findings generalize to the real world. We look at both generalizing to other people and generalizing to other situations. For our poster study, the participants were a convenient sample of undergraduates from a specific university. Can we confidently claim that these exact same results would apply to older adults or to kids in middle school? Now, what if external validity is poor? In experimental research, internal validity is almost always the top priority. To get absolute control over confounds, experimenters willingly sacrifice real-world representativeness. They will gladly run a study in a lab environment with a highly specific sample if it means they can confidently show a clean, unconfounded causal link. When we interrogate statistical validity, we're looking at the strength and precision of the data. We have to ask, is the difference between our group means statistically significant, or is it just a random fluke? We also focus heavily on effect size. This value tells us how far apart our group means are on the dependent variable, factoring in the spread or overlap of the scores. Look at the graphs on the slide. The distance between the means is identical across situations, but the effect sizes are completely different because of the spread of the scores. When scores are tightly clustered with less overlap, the independent variable had a larger effect. Finally, let's look at the main priority when evaluating a causal claim, internal validity. Whenever you are reading about an experiment, you must ask yourself these 3 fundamental questions about internal validity. Were there any design confounds? Did the researchers keep everything else perfectly constant so an outside variable didn't accidentally covary along with the intended independent variable? If an independent group's design was used, did researchers control for selection effects using random assignment or matching? If a within groups design was used, did researchers control for order effects by counterbalancing? If a study satisfies those three questions, well, then it can make a solid causal claim.